Good day, this is Professor Will, CBMD PhD. Today is Tuesday, January 30th, 2018, 2:29 p.m. Eastern Standard Time. Continuation, cell division. Among the many activities carried out by living cells, division is the most astonishing. A cell can divide into two cells, each of which can also divide into two, and so on through time, to create a population of cells called a clone. The discovery that genes are located in chromosomes was made in the first decade of the 20th century. Eukaryotic chromosomes are most clearly seen during cell division, when each chromosome condenses into a smaller volume. At this time, the greater density of the chromosome makes it possible to discern certain structural features. For example, each chromosome may appear to consist of two parallel rods held together at a common point. Each of the rods is an identical copy of the chromosome created during a duplication process that precedes condensation, and the common point called the centromere becomes associated with an apparatus that moves chromosomes during cell division. Chromosomes can be examined by using a microscope. Prokaryotic chromosomes can be seen with the techniques of electron microscopy, whereas eukaryotic chromosomes can be seen with light microscopes. Some eukaryotic chromosomes are large enough to be viewed with low magnification, 20x, others require considerably more power of less than 500x. In plants, development is less determinate. Tissues taken from part of a plant, for example a stem or a leaf, can be used to produce a whole plant, including the reproductive organs. Thus in plants, the distinction between somatic tissues and germ tissues is not as clear-cut as it is in animals. During animal development, a small number of cells are set aside to form the germline. All the gametes that will ever be produced are derived from these few cells. The remaining cells form the somatic tissue of the animal. In some creatures, such as plants, the germline produces both sperm and eggs. In other creatures, such as human beings, it produces one kind of gamete or the other. When a male and female gamete unite during fertilization, the diploid state is re-established, and the resulting zygote develops into a new organism. And many eukaryotic cells possess two copies of each chromosome. The condition referred to as a diploid state is characteristic of the cells in the body of the eukaryote. That is, the somatic cells, by contrast to sex cells or gametes, usually possess only one copy of each chromosome, a condition referred to as a haploid state. Gametes are produced from diploid cells located in the germline, which is the reproduction tissue of an organism. Most eukaryotic cells contain several different chromosomes. For example, human sperm cells have 23. The chromosomes of eukaryotic cells are also typically larger and more complex than those of prokaryotic cells. The DNA molecules in prokaryotic chromosomes and plasmids are circular, as are most of the DNA molecules found in the mitochondria and chloroplasts of eukaryotic cells. However, the DNA molecules found in the chromosomes in the nuclei of eukaryotic cells are linear. Good day, this is Professor Will, CBMD PhD. Today is Tuesday, January 30th, 2018. It is 1:53 p.m. Eastern Standard Time. Continuation, chromosomes where genes are located. Each chromosome consists of one double-stranded DNA molecule, plus an assortment of proteins. RNA may also be associated with chromosomes. Prokaryotic cells typically contain only one chromosome, although sometimes they also possess many smaller DNA molecules called plasmids. The shapes and activities of eukaryotic cells are influenced by a system of filaments, fibers, and associated molecules that collectively form the cytoskeleton. Other small membrane-bound organelles may also be found in eukaryotic cells. In animal cells, lysosomes are produced by the Golgi complex. These organelles contain different kinds of digestive enzymes that would harm the cell if they were released into the cytoplasm. Both plant and animal cells may contain peroxisomes, which are small organelles dedicated to metabolism of substances such as fats and amino acids. Ribosomes are found throughout the cytoplasm. Although ribosomes are not composed of membranes in eukaryotic cells, they are often associated with the system of membranes called the endoplasmic reticulum. The reticulum may be connected to the Golgi complex, a set of membranous sacs and vessels that are involved in the chemical modification and transport of substances within the cells. Good day, this is Professor Will, CBMD PhD. Today is Tuesday, January 30th, 2018, 1:10 p.m. Eastern Standard Time. Continuation. Some of the DNA within the eukaryotic cell is not situated within the nucleus. The extranuclear DNA is located in the mitochondria and chloroplast. We shall examine the structure and its function. Both prokaryotic and eukaryotic cells possess numerous ribosomes, which are small organelles involved in the synthesis of a protein, a process that we shall investigate. Individual chromosomes may be viable depending on when a cell is examined. In prokaryotic cells, the DNA is usually not housed within a well-defined nucleus. We shall investigate the ways in which chromosomal DNA is organized in prokaryotic and eukaryotic cells. The hallmark of eukaryotic cells is that their hereditary material is contained within a large membrane-coated structure called the nucleus. Within the nucleus, the DNA is organized into a discrete structure called chromosome. Algal and plant cells contain another kind of energy-recruiting organelle called chloroplast, which captures solar energy and converts it into chemical energy. Both mitochondria and chloroplast are surrounded by membranes. Eukaryotic cells are larger than prokaryotic cells, usually at least 10 times bigger, and they possess complicated systems of internal membranes, some of which are associated with conspicuous organelles. For example, eukaryotic cells typically contain one or more mitochondria, singular mitochondria, which are ellipsoidal organelles dedicated to the recruitments of energy from foodstuffs. All other organisms, plants, animals, protists, fungi are eukaryotes. The hereditary material, that is, the DNA, is not isolated in a specialized subcellular compartment. Organisms with this kind of cellular organization are called prokaryotes. Examples include the bacteria, which are the most abundant life forms on Earth, and the archaea, which are found in extreme environments such as salt lakes, hot springs, deep sea, volcanic vents. When we survey the living world, we find two basic kinds of cells, prokaryotic and eukaryotic. Prokaryotic cells are usually less than 1,000 millimeters long, and they typically lack a complicated system of internal membranes and membrane organelles. Some kinds of cells are surrounded by tough, rigid walls which are essential to the membrane. Plant cell walls are composed of cellulose, a complex carbohydrate. Bacteria walls are composed of a different kind of material called murin. Walls and membranes separate the contents of a cell from the outside world. However, they do not seal it off. These structures are porous to some materials, and they selectively allow other materials to pass through them via channels and gates. The transport of materials in and through the walls and membrane is an important activity of cells. Cell membranes also contain molecules that interact with material in cells' external environment. Such molecules provide the cell with vital information about conditions in the environment, and they also mediate important cellular activities. And cells are surrounded by a thin layer called a membrane. Many of different types of molecules make up the cell membrane. However, the primary constituents are lipid and proteins. Membranes are also present inside cells. These internal membranes may divide a cell into compartments, or they may help to form specialized structures called organelles. Membranes are fluid and flexible. Many of the molecules within the membrane are not rigidly held in place by strong chemical forces. Consequently, they are able to slip by one another in what amounts to an ever-changing molecule C. They also serve as energy sources. Proteins are the most diverse molecules within cells. Each protein consists of one or more polypeptides, which are chains of amino acids. Often the protein consists of two polypeptides. This protein is a dimer. Sometimes a protein consists of many polypeptides, that is, a multimer. Within cells, proteins are components of many different structures. They also catalyze chemical reactions. We call these catalytic proteins enzymes. Cells also contain nucleic acids, DNA, and RNA. Cells obtain energy when glucose molecules released from these chains are chemically degraded into simpler compounds, ultimately to carbon dioxide and water. Cells also possess an assortment of lipids. These molecules are formed by chemical interactions between glycerol and small organic compounds, larger organic compounds called fatty acids. Lipids are important constituents of many structures within cells. And carbohydrates such as starch and glycogen store chemical energy for work within cells. These molecules are composed of glucose and simple sugar. The glucose subunits are attached one to another to form long chain and polymers. Cells obtain energy when glucose molecules are released from these chains. are chemically degraded into simpler compounds, ultimately to carbon dioxide and water. Other kinds of molecules do not interact well with water. They are said to be hydrophobic. The inside of the cell, called the cytoplasm, contains both hydrophilic and hydrophobic substances. The molecules that make up cells are diverse in structure and function. The cellular environment. Living cells are made up of many different kinds of molecules. The most abundant is water. Small molecules, for example, salts, sugars, amino acids, and certain vitamins readily dissolve in water, and some larger molecules interact favorably with it. All these sorts of substances are said to be hydrophilic. In the early part of the 19th century, a few decades before Gregor Mendel carried out his experiments with peas, biologists established the principle that living things are composed of cells. Some organisms consist of just a single cell, others consist of trillions of cells. Each cell is a complicated assemblage of molecules that can acquire materials, recruit and store energy, and carry out diverse activities including reproduction. The simplest life forms, viruses, are not composed of cells. However, viruses must enter cells in order to function. Key points. Discoveries in genetics are changing procedures and practices in agriculture and medicine. Advances in genetics are raising ethical, legal, and political, social, and philosophical questions. But the impact of genetics goes beyond the material, commercial, and legal aspects of our societies. It strikes the very core of our existence because, after all, DNA, the subject of genetics, is a crucial part of us. Discoveries from genetics raise deep, difficult, and sometimes disturbing existential questions. Who are we? Where do we come from? Does our genetic makeup determine our nature, our talent, our ability to learn, our behavior? Does it play a role in setting our customs? Does it affect the way we organize our societies? Does it influence our attitudes toward other people? Will knowledge about our genes and how they influence us affect our ideas about morality and justice, innocence and guilt, freedom and responsibility? Will this knowledge change how we think about what it means to be human, whether we like it or not? These and other probing questions await us in a not-so-distant future. Such analyses are now routinely used in many situations to test for paternity, to convict the guilty, and to exonerate the innocent of crimes for which they are accused, to authenticate claims to inheritance, and to identify the dead. Evidence based on analysis of DNA is now commonplace in courtrooms all over the world. One way is economic. Discoveries from genetic research have initiated countless business ventures in the biotechnology industry. Companies that market pharmaceuticals and diagnostic tests or that provide service such as DNA profiling have contributed to worldwide economic growth. Another way is legal. DNA sequencing differ among individuals, and by analyzing these differences, people can be identified uniquely. Modern societies depend heavily on technology that emerged from research in the basic sciences. Our manufacturing and service industries are built on technologies for mass production, instantaneous communication, and prodigious information processing. Our lifestyles also depend on these technologies. At a more fundamental level, modern societies rely on technology to provide food and health care. We have already seen how genetics is contributing to these important needs. However, genetics impacts society in other ways too. However, medical geneticists have had some success in treating immune system and blood cell disorders by introducing the appropriate normal genes into bone marrow cells. To date, human gene therapy has made mixed results. Efforts to cure individuals with cystic fibrosis (CF), a serious respiratory disorder, by introducing copies of the normal CF gene into lung cells have not been successful. Human gene therapy is another way in which molecular genetic technologies are used to treat diseases. The strategy in this type of therapy is to insert a healthy, functional copy of a particular gene into the cell of an individual who carries only mutant copies of that gene. The inserted gene can then compensate for the faulty gene that the individual inherited. Without the added hormone, these children would be affected with Darwinism. Many other medically important proteins are now routinely produced in bacterial cells that have been transformed with the appropriate human gene. The large-scale production of such proteins is one facet of the burgeoning biotechnology industry. We shall explore ways of reducing human proteins in bacterial cells. Vats of these cells are grown to produce the insulin polypeptide on an industrial scale. Human growth hormone, previously isolated from cadavers, is also manufactured in bacterial cells. This hormone is used to treat children who cannot make sufficient amounts of the hormone themselves because they carry a mutant ally of the growth hormone gene. Molecular genetics is also providing new ways to treat diseases. For decades, diabetes had to be given insulin obtained from animals, usually pigs. Today, perfect human insulin is manufactured in bacterial cells that carry the human insulin gene. If a woman carries the mutant ally, she may be advised to undergo a mastectomy to prevent breast cancer from occurring. She may also be advised not to have children because each child would have a 50% chance of inheriting a mutant BRCA1 ally. The application of these new molecular genetic technologies therefore often raise difficult issues for the people involved. Advances in molecular genetics are providing new ways of detecting mutant genes in individuals. Diagnostic tests based on analysis of DNA are now readily available. For example, a hospital lab can test a blood sample or a cheek swab for the presence of a mutant ally of the BRCA1 gene, which strongly predisposes its carriers to develop breast cancer. Genetic diseases like the ones that Gerald studied are individually rather rare in most human populations. For example, among newborns, the incidence of phenylketonuria, a disorder of amino acid metabolism, is only 1 in 10,000. However, mutant genes also contribute to more prevalent human maladies, heart disease, and cancer. Classical genetics has provided physicians with a long list of diseases that are caused by mutant genes. The study of these diseases began shortly after Mendel's work was rediscovered. In 1909, there is also a concern that Bt corn might kill non-pest species of insects, such as butterflies and honeybees. Advances in molecular genetics have provided the tools and the materials to change agriculture profoundly. Today, policymakers are wrestling with the implications of these new technologies. Thus, Bt corn plants produce their own insecticide. The development and use of GMO has stirred up controversy worldwide. For example, African and European countries have been reluctant to grow Bt corn or to produce Bt corn grown in the United States. The reluctance is due to several factors, including the conflicting interests of small farmers and large agricultural corporations and concerns about the safety of consuming genetically modified food. Many corn varieties now grown in the United States carry a gene from the bacterium Bacillus thuringiensis. This gene encodes a protein that is toxic to many insects. Corn strains that carry the gene for Bt toxin are resistant to attacks by the European corn borer, an insect that has caused enormous damage in the past. Today it is widely used to augment the genetic material of many kinds of creatures. Plants and animals that have been altered by the introduction of foreign genes are called GMOs, genetically modified organisms. Bt corn is an example. Eukaryotic chromosomes duplicate when a cell's DNA is synthesized. The event which precedes mitosis is characteristic of the S phase of the cell cycle. And eukaryotic cells possess complex systems of internal membranes, as well as membranous organelles such as mitochondria, chloroplast, and the endoplasmic reticulum. Haploid eukaryotic cells possess one copy of each chromosome. Diploid cells possess 2 copies. Prokaryotic cells divide by fission. Eukaryotic cells divide by mitosis and cytokinesis. Cells, the basic units of all living things, are enclosed by membranes. Chromosomes, the cellular structures that carry the genes, are composed of DNA, RNA, and protein. In eukaryotes, chromosomes are contained within a membrane-bounded nucleus. In prokaryotes, they are not. The progression of eukaryotic cells through their cycle is tightly controlled by different types of proteins. When the activities of these proteins are disrupted, cells divide in an unregulated fashion. This deregulation of cell division may lead to cancer, which is a major cause of death among people today. The length of the cell cycle varies among different types of cells. In embryos where growth is rapid, the cycle may be as short as 30 minutes. In slow-growing adult tissues, it may last several months. Some cells, such as those in nerve and muscle tissue, cease to divide once they have acquired their specialized functions. The M phase in the cell cycle is a time when the mother cell actually divides. The phase usually has 2 components. One, mitosis, which is the process that distributes the duplicated chromosome equally and exactly to the daughter cells, and cytokinesis, which is the process that physically separates the 2 daughter cells from each other. Each time a eukaryotic cell divides, it goes through a series of phases that collectively form the cell cycle. The progression of phases is G sub 1, S, G sub 2, and M. In this progression, S is the period in which the chromosomes are duplicated, an event that requires DNA synthesis to which the label S refers. However, for these entities, the distribution process is not equal and exact. The mitochondria and chloroplasts are randomly apportioned to the daughter cells. The endoplasmic reticulum and the Golgi complex are fragmented at the time of division, and later are reformed in the daughter cells. And organelles, mitochondria, chloroplasts, endoplasmic reticulum, Golgi complex, and so on must also be distributed to the daughter cells. The division of eukaryotic cells is a more complicated process than the division of prokaryotic cells. After the chromosomes in a eukaryotic cell have been duplicated, the duplicates must be distributed equally and exactly on the daughter cells. At this rate, a single E. coli cell could form a clone of approximately 2 to the power 50 cells, more than a quadrillion. In just one day. In reality, of course, E. coli cells do not sustain this high rate of division. As cells accumulate, the rate of division declines because nutrients are exhausted and waste products pile up. Nevertheless, a single E. coli cell can produce enough progeny in a single day to form a mass visible to the unaided eye. We call such a mass of cells a colony. The mother cell's chromosome is duplicated prior to fission, and copies of it are bequeathed to each of the daughter cells. Under optimal conditions, a prokaryote such as the intestinal bacteria Escherichia coli divides every 20 to 30 minutes. A cell that is about to divide is called a mother cell, and the products of division are called daughter cells. When prokaryotic cells divide, the contents of the mother cell are more or less equally apportioned between the two daughter cells. This process is called fission. Within a clone, all the cells are genetically identical. Cell division is an integral part of growth of multicellular organisms, and it is also the basis of reproduction.